BioCentury This Week is brought to you by RemeGen Co. RemeGen is a leading biopharmaceutical company in China committed to discovering, developing, and commercializing innovative biologic drugs to address unmet medical needs in patients with autoimmune and ophthalmic diseases and cancer. RemeGen's flagship drug, Telitacicept, is a first-in-class BLyS-APRIL dual targeting fusion protein drug that has been launched in China for lupus and rheumatoid arthritis and is in late-stage development for various autoimmune diseases, such as Myasthenia Gravis, IgA nephropathy, and primary Sjogren's syndrome. RemeGen has research labs and offices across China and the United States.
It's springtime in biotech land, and that can mean only one thing. Medical meetings, of course, with biotechs presenting fresh takes on data, like so many tulips popping up in your CEO's garden. Well, today we are focusing in on two meetings. The American Academy of Neurology, or AAN, which wrapped up its annual meeting this month in San Diego, and the American Association for Cancer Research, or AACR, whose meeting kicks off in a matter of days. Now, for AAN, we'll be zeroing in on the Myasthenia Gravis space. It's a rare autoimmune neuromuscular disease characterized by muscle weakness. For AACR, we'll be looking at some first-in-human data and new targets. To discuss all of this, we have my colleagues Selena Koch, Executive Editor, and Lauren Martz, Executive Director of Biopharma Intelligence, here for this special edition of BioCentury This Week. I'm Jeff Cranmer, the other executive editor here at BioCentury, and I'm very pleased to say that today we have two special guests. We have Qing Zura. She is the Chief Development Officer from this month's podcast sponsor, RemeGen. And we have Amit Sachdev. He is the Director of the Division of Neuromuscular Medicine at Michigan State University. Go Spartans. He's also a PI on RemeGen's global phase 3 trial. Okay, let's jump right into AAN. There was a bit of Myasthenia Gravis data presented at AAN, some from RemeGen. They had a late breaker. We also saw some from J&J. And this is an indication that has really jumped into the spotlight in the past couple of years because of a company from Europe, argenx. Argenx's drug for myasthenia gravis has really taken off and has done quite well in the market. What we want to talk about today is, well, what's next? What's coming up next? And Selena, I know you have been digging into the space. You've edited a lot of stuff by our buddy who is hopefully fast asleep in the UK, who follows argenx quite closely. That's Stephen Hansen. What are you seeing, Selena?
Well, maybe a good place to start is with the class of compounds that argenx's Vyvgart is a member of. So argenx clearly has first mover advantage here, which they're making good use of. But FcRn, this is a neonatal Fc receptor, which is a bit of a misnomer as it's expressed throughout life. But the idea behind it is that it blocks the interaction between the receptor and IgG usually in acidified endosomes. So normally FcRn binding to IgG would rescue it from lysosomal degradation, allowing it to be recycled back into circulation. So when you block it, you get depletion of IgG, and that depletion includes the pathogenic autoantibodies that drive the disease. Although, it's worth noting, it's not specific to them. So at AAN, J&J presented a program update, really, on its molecule, which is also in this class. So nipocalimab is actually under FDA review right now, under priority review. It's another one of these compounds. There was also data there from a late-stage program from Amgen, a CD19 mAb called Uplizna. Beyond those programs, we also saw some data unveiled from RemeGen, which has a different mechanism. It has two targets that are kind of gaining in popularity in a variety of autoimmune conditions called Bliss and April. Maybe Lauren wants to jump in and talk a little bit about those as she's been following I&I and new targets.
Sure. Thanks, Selena. Bliss, April, and the receptors that these targets bind on B cells have been emerging as sort of a new class, a way to go upstream, as we may hear about later from some of our guests, of the complement pathway, FcRn, all of the autoantibody, the IgG targeted mechanisms. So in this case, Bliss and April are the ligands that target a few different receptors on B cells. B cells are behind several, are pathogenic and produce autoantibodies that are a problem in a range of autoimmune diseases. And so we've seen a lot of different ways to sort of target the B cells themselves, prevent them from making the autoantibodies that become a problem. These targets have been increasing in popularity for those B cell mediated diseases, lupus, and things like that. So MG is a logical next step. I think we're seeing some very positive data from the RemeGen program and a bit of an explosion in the pipeline of therapies against these ligands and the receptors that they target.
Maybe good to talk about what are the benchmarks to beat, right? So if you look at this class, I think there's 3 FcRn inhibitors across their trials. They're measuring often responder rates, which I think have been in the 50 to 70% range. Presumably pushing that up higher would be good. Some of the other treatment goals, lengthening out that interval between doses, or maybe in the case of something like a CAR T, maybe you hope for one and done. How are you thinking about these different kinds of modalities and targets, Lauren, in the context of that kind of remaining need?
Yeah, I think there are a few different goals. You've talked about some recent biologics that have been approved for the indication. Not everyone responds. So having a breadth of different mechanisms, first of all, is very important. The trend for disease areas within I&I, and this straddles I&I and neurology, we have some effective mechanisms that work for many patients. These are biologics that require frequent injection. So as you said, oral therapies are a trend. Expanding the interval between dosing is a trend. And then there is that long-term vision of potentially resetting the immune system, which seems like it's something that may be possible with B cell targeted strategies, particularly those that can deplete the B cells, not only in circulation, but deep within the tissues, which this may be an indication where that's possible. And that's something that many different groups now are testing in the clinic in future clinical trials. I think it's wide open, the potential in this space.
And maybe we'll say just one more quick thing before we bring in our guests here. There does seem to be a need also, I guess you could call it a precision medicine need, but not everybody's disease is caused by the same antibodies, right? A lot of times they're antibodies against the type of acetylcholine receptor that exists in neuromuscular junctions, but sometimes they're against other self-antigens that cause a similar phenotype. So expanding out the phenotype of patients would also be helpful.
Excellent. Well, hey, let's bring in the first of our 2 special guests. We're very pleased to have Dr. Amit Sachdev. As I mentioned at the top, he's director of the Division of Neuromuscular Medicine at Michigan State. He is a PI on many, many drug trials. And in particular, he is a PI on RemeGen's global phase 3 trial for Telitacicept, which is also known as RC18 in this space, Myasthenia Gravis. Amit, welcome to the podcast.
Thank you so much for having me, Jeff, Selena, and Lauren. I have enjoyed listening thus far to this wrap-up. Very on point.
Excellent. Well, I'd love to hear your take about the current landscape of what's in the pipeline. What are you watching? What's coming up next?
Yeah, well, we have the pleasure of caring for about 150 patients with myasthenia gravis here at Michigan State. And what's very clear is that there is unmet need for this patient population. And that need comes in the form of different ways to treat the disease, but also access to those treatments.
Yeah, I would love to get your insight on how the kind of therapeutic profile of Telitacicept maps onto the remaining patient needs.
Yeah, thank you, Selena. So many providers in neuromuscular medicine will follow the guidance from the Myasthenia Gravis Foundation of America on managing patients with myasthenia gravis. And that guidance starts with prednisone or other corticosteroids. And the second agent on is off-label use of an oral T-cell inhibitor. Usually that's going to be mycophenolate or azathioprine or cyclosporin. What's interesting about those oral T cell inhibitors is how high they act. And there's no question there's good data that they do effectively manage disease, but they take a long time to be effective. There is black box warnings on a number of these drugs, and those black box warnings include risk of malignancies. And so there's clearly a level of comfort with managing the disease with very high level treatments. And there's an absence in the market for doing that in a more refined way.
So it sounds like, one of the goals would be kind of sparing patients treatment with steroids or other sort of blanket immunosuppressants of yesteryear. Is that the way these trials are designed?
So in myasthenia gravis, there has not been a major focus on steroid reduction or sparing, not at least in the current generation of treatments. The very first generation of clinical trials did focus a great deal on reduction of steroid use. And I think most neurologists today are not using the 1 milligram per kilogram dosing that we used to use to manage these diseases. And so they are using a combination of treatments these days, but a lot of patients are still experiencing moderate degrees of disease burden, and there's underemployment in this patient population. And so refining how we target disease, I think, is pretty important. It allows us to better treat disease, and it allows us to really tailor what risks we're asking the patient to take.
Maybe tell us a little bit about what I guess you're most hopeful for with the sort of product profile of Telitacicept.
The ideal intervention in myasthenia gravis is an intervention that is durable and an intervention that doesn't require a high burden of administration. So these are really the two things that we look for. The treatment approach with this clinical trial program is attempting to achieve those things. We'll have to see whether or not it can. Durability in particular is really important. Patients with myasthenia who have an exacerbation are liable to end up in the hospital, and we want to avoid those hospitalizations and exacerbations if we can.
What is the dosing schedule?
The dosing is subcutaneous injections each week.
Amit, could you please tell us about the new data that were presented at AAN?
Thank you for that question. Now, I do only have access to the poster that was presented at AAN, and I have had a chance to take a look at it. I will say the outcome measure that the study used, the MG-ADL, is the same outcome measure that has been used by the majority of studies in this era. And the magnitude of effect, roughly 5-point reduction in MG-ADL, was a little bit better than I have seen in some of the other studies. And I find that to be encouraging. It's one of the reasons we are comfortable being a phase 3 site for the global study.
All right, thanks for that, Amit. I'd like to bring Dr. Qing Zura into the conversation now. She is the Chief Development Officer of RemeGen, sponsor of this drug. Qing, welcome to the podcast.
Well, thank you very much, Jeff, Selena, and Lauren. I think this discussion is very important in the treatment of patients with myasthenia gravis. I think at RemeGen, we are very excited about telitacicept, known as RC18. So the drug is already approved in China since March 2021 for the treatment of lupus, and then subsequently have it approved for the treatment of rheumatoid arthritis. And the phase 3 result presented at the AAN, as Dr. Sachdeva just described the result, we are encouraged, and we look forward to our global phase 3 studies, which will involve 14 countries in the study. And also for the phase 3, the China study, we only presented in the first part of the study, which is the 24 weeks double blind. And we look forward to present the open label part, which is a part B of the study. And that gives the patient additional 24 weeks treatment. I think to Dr. Sachdev's point of durability, so we're looking forward to share the data in the future meetings, and that will provide some data in terms of long-term durability to treat a patient with myasthenia gravis and also the safety profile. So which is consistent with what we have seen in all these autoimmune diseases that we have been doing clinical trial with. For example, the lupus, the RA, Sjogren, and IgA nephropathy.
Jing, can you tell us a little bit about the patients you're enrolling? Are they kind of across the board in terms of the type of self-antigen that's driving their disease?
Yes, that's a very good question. So we're currently enroll patients who are AACR positive or mask positive.
All right, and I know that the global trials are in phase 3, and it looks like your BLA is in the hands of China's NMPA. Is that correct? When are you expecting a decision?
Yes, so the China regulator, what we call the CDE, right? They have accepted our submission. So we're actually really pending approval. So we're very excited looking to see what next step.
Sounds good. And CDE, of course, is the Center for Drug Evaluation at China's National Medical Products Administration. Well, I'd like to thank both of our guests so much for joining us to talk about the latest in myasthenia gravis, especially coming out of this year's AAN meeting in San Diego. Amit, Ching, thanks for coming on to the BioCentury This Week podcast.
That was fun. Thank you very much.
Thank you for having me.
Alrighty, we're going to go to a quick break.
BioCentury This Week is brought to you by BioCentury Grand Rounds. Grand Rounds. BioCentury's R&D conference will return in 2025 to the US and Europe to debate key bottlenecks in translation and unlock scientific breakthroughs with commercial potential. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore cutting-edge advancements in early-stage R&D as we dive into disease biology, therapeutic modalities, and enabling technologies. Join us in Chicago this June after ASCO for the second BioCentury Grand Rounds US, and in Cambridge, UK this September for the inaugural Grand Rounds Europe. Register and learn more at biocenturygrandrounds.com.
And we are back. A special thanks to our guests, Dr. Zura from RemeGen and Dr. Sachdev from the Michigan State University, who is a PI on RemeGen's global phase 3 for Telitacicept. Okay, let's turn to AACR. It's one of the conferences we follow super closely here at BioCentury every year. Lauren, it's sort of the delight and, dare I say, bane of her existence when the AACR abstracts come out by the thousands, and Lauren loves to pour through them to see what first-in-human data is next up, any cool new targets she has spotted. She's been doing this for years now. So let's bring her in. Lauren, what have you noticed in digging through the abstracts?
Thanks, Jeff. Yeah, so we're using the first-in-human clinical trials and the new targets that are included in the AACR abstracts this year as two ways to get at the translational trends that are sort of at the forefront. This is the innovation that's moving into the clinic, that has just moved into the clinic, that companies have chosen to present in these abstracts and that the conference has chosen to accept. And then these are targets that BioCentury hasn't seen addressed by marketed products or any products that are of the same modality for the same disease area that are more than a couple phases ahead in the clinic. So new targets. Within the first in human set of abstracts, we found 44 this year. The big trend was bispecifics. I feel like a bit of a broken record because everything is bispecifics in 2025, and this meeting was no exception. Compared with any other new modality and some of the traditional modalities, there were at least 3 times as many bispecific products that are being highlighted in the first in human studies this year. What I think was interesting in those is that just 2 of them were sort of the traditional CD3 targeting T-cell engaging bispecific that, that we've been following for a decade. These are a combination of different structures. There are multi-specifics, trispecific antibodies that engage T cells or NK cells. There are other T cell engagers that bind a different target. I think that it's just 4-1BB in these studies. And then there are a bunch of abstracts that are kind of getting at the Akeso model of using a bispecific structure to target simultaneously 2 different cancer mechanisms.
And localize them to a cancer cell in a way that minimizes tox? Is it a different kind of value in these scenarios?
In some cases, I think that's the idea. So what I've heard is that bispecific design in cancer, it's all about creating new biology. So localizing the effect of maybe a cancer mechanism that's too toxic. Some of these are bispecifics that engage or inhibit 2 different immune pathways, trying to have a stronger immunostimulatory effect on the cancer cells is another thing that we're seeing a lot.
Yeah, combining, immune mechanisms, either taking 2 breaks off or taking a break off and putting on the gas. These ideas have been around for quite a long time. What feels fresh about this current crop of abstracts?
So there aren't too many examples, but it's different targets than we've seen before. Let's see. An example is a program from Sparks Biopharmaceuticals, which is targeting PD-L1, the T-cell checkpoint ligand with one arm, and then LILRB2, which suppresses the function of B cells and myeloid cells. So, it's not just 2 T-cell checkpoint targets, which we've been seeing for a while. This is trying to engage the different parts of the immune system in a single therapy.
Well, there's no doubt we're going to be continuing your broken record of talking about bispecifics many more times this year, but I don't think bispecifics were the only new modality that were kind of standing out in the meeting abstracts. What about targeted protein degradation?
So targeted protein degradation is something that showed up quite a bit in the new targets analysis that we did. The promise of targeted protein degraders, the molecular glues, the PROTACs, things like that, is that you could potentially get at targets that small molecules, traditional small molecules, can't necessarily inhibit very well, because in this case, you're degrading them rather than trying to find a way to block their activity. What we've seen in terms of early clinical data for the modality is a lot of validation with established targets. What we're seeing now, I think this is a good reflection of that, is this promise of opening new target space, because there are, within the limited number of new targets that we found, there were 3 new proteins that could be degraded by a PROTAC type structure that we haven't seen before. And one of the abstracts that we found also defines a new E3 ligase that could become part of that PROTAC structure, part of the machinery that actually makes up the therapeutic.
Well, maybe even earlier stage, I'm not sure, but a kind of a new area of biology I think you've noticed coming to the fore is around glycosylation-related targets. Can you tell us a little bit about that?
Yeah, so I don't think this is necessarily a new space, but it's something that we're seeing more and more of, in the literature. It's been featured in our distillery analysis quite a bit. It's also something that we're seeing in the abstracts is new targets that are involved in the glycosylation process of proteins on the surface of cancer cells. There are a few different ways that glycosylation contributes to drug resistance in cancers. Glycosylation of PD-L1, for example, helps that ligand stay on the surface of a cancer cell and helps that evade immune detection. There are also cases where too much glycosylation of different targets can actually block the binding of a targeted cancer therapy to that target on the tumor cell. I think there's an increasing understanding that this is one way that cancer cells can resist immunotherapies. It's been known that it's one way that they can help resist targeted therapies as well. And there were quite a few. I think there were 4 targets that were glycosylation related or related to other post-translational sugar modifications on the tumor cell surface.
Thanks for that, Lauren. Your article previewing AACR up now on biocentury.com, biocentrypodcast.com, and via the link in the show notes. And for those of our listeners who are at AACR, we wish you a very good conference and much success with your posters, your presentations. And we will be back with our regular podcast on Monday. Thanks for tuning in to this special edition of the BioCentury This Week podcast. Kendall Square Orchestra provides the music for BioCentury This Week. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.
BioCentury would like to thank RemeGen for supporting the BioCentury This Week podcast. To learn more about RemeGen's products and pipelines, you can go to www.remegen.com.